Welcome to Cryo-Talk, a bite-sized bio podcast sponsored by Thermo Fisher Scientific, featuring conversations between your host, Eva Amsen, and experts in the field of cryo-electron microscopy.
Today on Cryo-Talk, we are joined by Yiorgo Skiniotis, Professor of Molecular and Cellular Physiology, Structural Biology and Photon Science at Stanford University. We're talking about his research career.
I was always curious as to how we can bring different methods sort of together.
Catching up on classic literature.
These are the days that I'm grappling again with James Joyce.
The surprising job he would choose if he wasn't a scientist.
I often fantasize about being out there and catching fish.
And the importance of cultivating diverse research interests.
None of us wants to do the same thing over and over again. It becomes very boring.
All in this episode of Cryo-Talk. Hi, and welcome to Cryo-Talk. I'm Eva Amsen, and I'm here today with Yiorgo Skiniotis, Professor of Molecular and Cellular Physiology, Structural Biology and Photon Science at Stanford University. His research group uses cryoEM and 3D reconstruction techniques to study macromolecular complexes in cell signaling, such as transmembrane receptors. Yiorgo, how are you today?
I'm good. Thank you very much for having me.
Yeah, thanks for joining me. So, as I told people, you're at Stanford now, but can you tell me a little bit about your research background? So, what brought you there?
Oh, what brought me there? Life science. So I grew up in Greece. I studied biochemistry at the University of Leeds in the UK, and then I did 2-month sort of diploma thesis that was actually on electron microscopy, 2D crystallography of all things, and sort of that level of direct imaging that an electron microscope would provide you compared, let's say, to other structural biology techniques, sort of piqued my interest. And it was one of the techniques that was definitely not as much in fashion as it is now. So I did my PhD at EMBL in Heidelberg, at the European Molecular Biology Lab. And I actually did cryo-EM of kinesin microtubule complexes, trying to understand how kinesin motors change their conformation as a function of their nucleotide state, and that enables sort of a walking mechanism on the microtubule. And then it was pretty clear at the time that, that was obviously helical reconstructions, because we were trying to select microtubules that you could apply helical averaging. But it was clear that the next thing was actually, or it wasn't, I shouldn't call it next, but it was still in the earlier stages of development. It was single particle analysis, right? Without having the need for symmetry. For example, in going after individual molecules and averaging them. So I, decided to do a postdoc at Harvard Medical School. I was actually in Tom Valtz's lab, and there I learned single particle cryo-EM. I spent most of the time there to actually do negative stain analysis.
Oh, wow.
Because the majority of the targets, so I had done cryo-EM for my PhD, but for most of my postdoc, I actually did negative stain, because the targets that I selected were very, very small, like receptors, for example, small signaling cell surface receptors. But that also, negative stain is also a bit of an art, right? It has a lot of caveats, but it's art in imaging, and you can extract very useful information. And then I moved to the University of Michigan as an assistant professor. I built a research program around signaling receptors, cytokine receptors at the time. Then we started slowly going after G protein coupled receptors. I got tenured in Michigan, and then I, moved to Stanford about 5 years ago.
Wow. And how are you liking it there so far?
I like it. Stanford is an interesting place. You can't beat the weather,
Yeah. I mean, I'm in London, so I I can't imagine good weather. Yeah, so one of my questions, which you actually already answered, was how did you get started with cryo-EM? So I guess you started during your PhD.
I started during my PhD, and it was a very interesting time. I mean, I have to say it was one of the most fulfilling times that I ever had. My background is actually in biochemistry, right?
Mhm.
So when I chose a cryo-EM project, I sort of had to do everything from A to Z, from the molecular biology, the protein biochemistry, learn cryo-EM, understand many of these concepts that at the time were somewhat esoteric, and often the doubt of disputes resolution and what have you. But I also feel that this prepared me well for what was to come, because I think I'm fortunate in that I developed a sort of more well-rounded approach to a structural biology project, right? Which is not just the electron microscopy, not just the image processing, but perhaps even more importantly, the target preparation, right? What are you looking at? How do you purify it? What state is it when you purify it, and how you can fine-tune the system that you're interested in order to get, reasonable information.
Yeah, because what are some of the advantages that you found of cryo-EM when you're looking at receptor structures in particular?
Well, so I started looking into cytokine receptors, and these receptors have sort of a series of Ig-like domains, to put it simply, small round domains that are connected like beads on a string. So when you actually purify them and you take a look, you realize that these are very flexible assemblies. And this is the reason why, besides some small parts of these molecules that were determined by X-ray crystallography, this flexibility essentially hindered applying crystallography on the full ectodomains, so the entire receptor in complex with its ligand. So the advantage of single particle cryo-EM is essentially that you don't have to go through crystallization, and you can obtain average information from different classes that show distinct conformational states. And that, of course, is also crucial to understand biology. Things are not static. Things are moving left and right and up and down, because they need to execute a certain type of work. So this is the true power of single particle EM, which I would argue it probably has not been fully realized yet.
That actually brings me to my next question, which is a little bit about the future. So you're also director of Stanford Cryo-EM Center, so I'm sure you've got a bit of an overview of how cryo-EM is being used beyond your own work. Now, if you think like 5 to 10 years in the future, what are some of the exciting applications that you think we can expect of cryo-EM?
Yeah, well, I'm afraid I'm not going to be able to tell you anything new here. I think Joachim in the first podcast that you hosted talked about time-resolved cryo-EM. This is definitely one of the next frontiers, and our approaches are at their infancy. I would argue that cryo-EM, single particle cryo-EM, is still not used to its full capabilities. And what I mean by that is that we're still going after one or two structures at high resolution, And we don't take full advantage of the ability of the technique to look at the entire ensemble, right? Because these are single particle, single molecule measurements, if you wish. Part of the problem is that our approaches to the whole sort of methodology come from crystallography, where we try to purify as well as possible macromolecular complexes in a single state, right? But the question is, what would happen if we start in a controlled way, of course, inducing states, and instead of going after steady state samples, let's say samples that are all in the same state, we see them in flux. And then we take advantage of these wonderful sort of classification methods that have been developed much more rigorously in the last few years to actually see how things move, the entire dynamics, and how can you go from A to, E, and seeing all the intermediate states, B, C, D, for example. So time-resolved cryo-EM is definitely one of the next frontiers. Of course, looking at more complex assemblies that somewhat recapitulate better, if you will, what happens inside the cell is definitely one of the next frontiers as well. And what I mean by that, let's take, for example, our own work on G-protein coupled receptors. You see hundreds of, you have seen hundreds of structures right now, published, and this is a GPCR and a G protein, but a GPCR and G protein are not there on their own. They're downstream effectors, there is a membrane environment, there are additional players into this whole system. So the question is, can we sort of in vitro recapitulate higher order assemblies that give us a more pragmatic view, if you will, of how these things work? And of course, this is also the connective tissue with the other big player, which is tomography, cellular tomography, looking at macromolecular complexes in situ. And there's definitely a lot of development that is required in that space, particularly when we are targeting complexes that are beyond sort of the model complexes, whether it's ribosomes that are very easily distinguishable and you might have a lot to average, or proteosomes or what have you. How can we go after lower occupancy targets that are also more difficult to recognize? And this is where you're having the interplay with fluorescent microscopy and doing correlative cryo-electron microscopy, light microscopy. So there's going to be an explosion of new developments there in terms of how do we even go in a rational way after such targets?
Yeah, that's going to be very exciting once we get to that point. That's
yeah,
because of course in biology everything is dynamic. It's not just a snapshot of one or two molecules at a time. I guess speaking of dynamics, let's talk a bit about your own career and research again. Has there ever been a moment where things took a surprising turn for you in your career?
I have to tell you, there was never really an aha moment that you wake up one day and you're like, I have to change direction completely. My positioning in all this, or my view is, I'm trying to tell a story, right? And all these stories interconnect, and one story takes you to the next. And the plot is very often very difficult to sort of plan ahead.
Yeah.
You can plan the next couple of steps, but suddenly, especially as, let's say, you become a little bit more senior and you have been involved for a number of years in many different projects, then you start drawing connectivities between all these different plots. And this is where the new thing comes, essentially. But yeah, to give you sort of as straight answer as possible, I never had like, oh my God, I have to change completely direction. You hone your skill, yeah, right, slowly, and you see again how all these connectivities build and how you can bring additional. I mean, I always approach things from a more holistic perspective, if you wish. And I was always curious as to how we can bring different methods sort of together and see how they communicate with each other and tell part of the story. Whether it is cryo-EM and molecular dynamic simulations or more sophisticated biochemical experiments, and how can we go after dynamics with true single molecule biophysical measurements, let's say FRET, for example.
It sounds like you're pretty in control then of like what's happening next. So what do you think will be next for you in your career over the next few years? Do you have any exciting plans?
Well, I never said I was in control about
it sounds like you are.
What is happening next. I'm just, I guess through the years, I have become a little bit more paced, if you wish, with what I'm doing. Part of being a scientist is also having, developing an intuition and also the quick reflexes. If there is something interesting to follow it up, and still I'm convinced that there's many things that we kind of leave behind because we don't have the capacity to focus on everything, right? I'm not quite sure about the next steps in my career now that cryo-EM has become much more mainstream. Maybe it's time for me to do something else. I don't know.
Do something very surprising. Yeah. And then the next time you're on a podcast and I ask you about a surprising change in your career, you can talk about that. Yeah.
Well, I guess what I'm trying to tell you though is that, whatever changes we decide to do are never like always smooth. As every scientist, you struggle with things. You try to figure out what is worth doing. How is it worth spending the next 10 years of your life in this particular space, in the scientific space? What questions are worth asking, right? None of us wants to do the same thing over and over again. It becomes very boring. And yeah, I'm not quite sure. It's something that I'm grappling with on a regular basis. What is worth doing and where should I go next, right? And there are periods that I'm much more calm about what I'm doing, and there are other periods that I just want to change everything around me.
It's like that for me. I don't like doing the same thing over and over.
Yeah.
And I noticed a bike behind you. Is cycling one of your hobbies?
Cycling is on my hobbies. I used to cycle much more indoors, especially at the start of the pandemic. But quite frankly, the weather is so nice around here that it was silly. I found myself staring for too long in front of a screen, cycling virtually. So I haven't used it in months, to be honest with you. I'm just biking outside.
And what else do you like to do for fun?
Yeah, it's a good question. I mean, it sounds mundane, but it's like, I watch the occasional movie and catching up with cinema that is not the cinema that it used to be. I grew up with, European cinema and movies, and I'm somewhat disappointed about what has been happening more recently. I'm not big into cooking. I suggested that you might ask me something.
Yeah.
This line. I like cooking for other people. I would like having friends around, and of course the whole pandemic phase limited quite a bit this option. I like traveling. I travel. I started traveling again, and I'm just like a kid because we've all been so confined for the last couple of years that now that traveling is opening up again, it just makes me very, very happy. And I spend time with my kid. That is my joy.
Mm-hmm. That all sounds really nice. Yeah, traveling. Everyone has been traveling this year. I don't know if the airline industries can keep up with it, but
they can't.
Nope. So yeah, you mentioned that you love films. Do you have anything you can recommend us? Anything we need to watch?
I did watch, and you can watch it on Netflix actually. I did watch The Power of the Dog by Jane Campion, and I thought it was exceptional.
That's on my to watch list. I still need to see that. Yeah. I've got a couple of other quick questions for you. Do you prefer the city or the countryside?
Oh, I'm of two minds. I grew up in Athens, and I think, it's a chaos of a city, but once you know it, is an amazing environment, right? You have so much input and different kinds of influences. And in a way, I've been thinking about this about my kid, because I would also like him to have the experience of the city. But I feel that I have also reached the point in my life that I like visiting cities. I don't necessarily need to live in them, because I enjoy, I live very close to campus, and here is just like, I don't know, it's very quiet, it's very green, there is no traffic. You don't stress with anything else other than work, right? So I've learned to appreciate that. I've learned to appreciate the easiness of living in such an environment, more countryside, if you will. But I always love visiting cities and tasting what they have to offer.
Yeah, and you're close to a city, so you can just
go
whenever you need to. Yeah. I also see a bookcase behind you, and I can't see what's in it, but do you have any books you can recommend us?
Oh, God. My perspective about books keeps on oscillating, right? There are books that I adored as in my 20s that I couldn't look back again, later on. So it's something that keeps on evolving. I definitely don't read even half of how much I used to read. I feel that I'm doing a return though. I'm going back to more classical content, if you wish. So these are the days that I'm grappling again with James Joyce, that I think is not going to be to the taste of the majority of your audience.
Not light reading.
Yeah.
And do you like music?
I like music, but not in a very sort of targeted way. I operate better with music on the background, but as long as it is not something very offensive to my ear, then I can listen to many different styles.
I guess background music is
I mean,
Could be anything.
there's always a mix. There is always a mix between, you know, I could listen from, you know, Iron Maiden to Depeche Mode, go to something much more jazzy. Again, you know, it's not about the style of music. I think its particular style has certain things to offer depending on how you feel and the time of the day. But usually I try to have music around. It keeps me calmer.
Mm. It's nice to have some sound in the background.
That's right.
And this is always a hard question for, well, maybe you've thought about it before, but if you're not a scientist, what would you be? What is your alternate reality career?
If I was not a scientist, I think I would be a fisherman.
Oh, why?
Many different reasons, actually. So I grew up next to the sea. My parents were islanders, so I used to spend many summers on islands. Mm-hmm. And I've grown this sort of very deep connection to blue, mm-hmm. to the sea, salt, the arid landscape of the Cyclades complex where my parents came from. And being a fisherman is a very, very tough job, but it's also a job that you're always out there. Yeah. And there is a lot of science behind it. There is a lot of science and the skills and the tricks. Like I was noticing as a kid, you know, these people, they were really good fishermen, and there's some horrible fishermen, and then everyone else in between. But the good fishermen really had a system. I mean, they were thinking about things. It wasn't just instinct or experience, right? They knew where to go. They were doing their calculations, and they had the ability to be out there and not be sort of enclosed within four walls. So, you know, that's because as all of us were spending a lot of time in front of a computer, I often fantasize about being out there and catching fish.
That sounds amazing now. Now I want to go outside and fish. And finally, do you have any advice for early career researchers?
You know, I have a very close friend, Roger Sunahara, who is a professor at UCSD. And, you know, at the bottom of his email, he has this saying, just go crazy and do it, or just get crazy and do it. And, you know, I think, you know, the usual advice is be persistent and go after your dreams. You know, my advice is like, just get crazy and do it. You know, life is short. Experience, go after new experiences and see what comes. Stop calculating everything.
That's amazing advice. And on that note, thank you so much for joining us today. That brings us to the end of today's episode. So thank you, everyone. Yeah. Thanks everyone for listening or watching Cryo-Talk today, and have a great day, Yiorgo.
Thank you. Bye-bye. Thank you.
Bye.
Thank you for listening to Cryo-Talk, a bite-sized bio podcast sponsored by Thermo Fisher Scientific. To view all audio and video recordings from this series, please visit bitesizebio.com/cryotalk.